Tamer Mohamed, and he's going to talk about novel approaches to endogenous heart repair. And Dr. Tamer graduated from the University of Manchester, and he did his postdoctoral training at the Gladstone Institute. His patents have actually established a company that's now worth over $50 million of investments. And so he's had that translational side as well, and his focus is on cardiac repair. His manuscript has been featured on the cover of Cell, which is a very, very prestigious journal if you didn't know. And so with that, I'll turn it over to Dr. Tamer.
Yeah, thank you so much for the introduction, and thank you for inviting me to give this seminar. So this is probably more extreme biology, so I'm trying to make it as simple as I can. I'm happy to be interrupted in the middle if anybody has some questions. So basically what we've been developing over the past few years are novel therapeutic approaches for heart failure, to repair the heart from inside, so within inside. So basically we are using whatever in the heart to repair itself. So just to give you a background about what heart failure is. So if somebody gets a heart attack, so due to eating too much fats or deposition of fats in some arteries, like what you see here in this. So if the fat deposits in some of these coronary arteries, it blocks these arteries, and these arteries are carrying the blood to the heart muscle. So this is what feeds the heart, basically. So if you deprive the heart from the food or the blood, so the heart muscle starts to die, and it's like healing process. So if you get injured in your skin, it heals and it makes like a scar. So that's what happens basically. So the muscle part of the heart dies out, and then it forms like a scar here, and most of the muscle cells are gone. And now you're left over with some solid tissue, fibrous tissue, like what you see on your skin when it gets healed after injury. So the way to fix this problem or to make the heart getting better, so by time the heart loses its ability to pump the blood to the whole body afterwards. So to fix this, we need to add more muscle cell to this part at least, or to support this part, right? Like when your skin heals, so you get the scar out and you get some new cells getting in to replace your injury, right? Otherwise, the heart will fail to pump blood into the whole body, and then death will occur. And one of the major causes of death, of course, is heart failure. So it took us a lot of time to understand how could we get to the process where we can fix the heart using its own heart, its own cells, basically. So one of the major developments that we in the field, basically in the past 10 years or so, is we started to understand how we develop, the development. So from fetal stages up to after birth and up to adulthood, and how our basically ability to regenerate or to get our cells healthy is deteriorating from the fetal stage up to you go adult, and then you go over 60 and over 70 and so on. So at least in the heart field, in the past 10 years or so, we developed the knowledge that in fetal stage, so in the mother womb, the heart is able to regenerate itself. So these muscle cells are able to divide, and if you make injury to a fetal heart, the heart heals very well and the muscle cell reproduce again. And basically you lose this ability of regeneration just after birth. So few weeks after birth, you lose completely this ability. And by adulthood, you completely has no ability, or the heart cells or the muscle cells on the heart are not able to proliferate or to heal anymore. So so that basically if we can reverse this process, so go back from this muscle, from this cell to this cell, basically I think we can make the heart able to regenerate again, right? Okay, so how to do this? I think it needs a lot of understanding of how this process happens so we know how to reverse it back, right? So I think one of the major understanding that helped us to figure out this is our understanding of development again, and how we all start from one stem cell. So every one of you knows that everybody or any human being started from only one cell, and this cell started to develop a blastocyst, and then these bunch of cells start to differentiate to different organs and different types of cells. So how this process happens is what controls this process. So you start with the same cell and you get multiple different cell types out of this single cell. So we started to understand a bit about this by understanding that this single cell develop a blastocyst, and this blastocyst get differentiated into 3 major layers. And these major layers are Ectoderm, Endoderm, and Mesoderm. There's another layer which is the germ cells, but this is not very important. But these are the 3 major layers, the Endoderm, Mesoderm, and the Ectoderm. And each of these layers is responsible about making certain tissue types. Okay? So for example, for the heart specifically, the Mesoderm is what makes the heart. The Ectoderm makes the neuron, the skin, and the ligament cells. The Endoderm makes your gastric and pancreatic cells. So how this happens, I think this was the next step. So what controls the process of a single cell to become 2 different cells, or 2 different types of cells? Even though every cell has got 46 chromosomes, they're encoding the same DNA. So what makes the cells different? So one major breakthrough is we found that the answer to this question was the epigenetics. So there's a control on the DNA that happens, and this control on the DNA is called either methylation or acetylation, and according to these methylation or acetylation in certain sites of the DNA, this can block the expression of certain genes or activate the expression of other genes. And according to what genes are expressed in certain cell, it will make it specialized in doing this job. So for example, the heart cells are producing some muscle fibers and so on. So so for the cell to become a heart cell, somehow there are some factors, they are called methylation and acetylation enzymes, that comes to the DNA and start to methylate at certain sites, and these certain sites are controlling the expression of these myofiber genes. And by controlling the expression of these myofiber genes, this cell becomes specialized to be cardiomyocyte. And another cell, there will become this methylation machinery, and it will methylate sites that control expression of absorbing villi or something in the gastric cells or something like this. So basically we found out that this methylation is the control for this whole process on how the cells get specialized. And this opened a lot of discoveries afterwards that we could basically convert, if we can control the methylation, we could convert one cell type to another cell type, right? So we can basically have a skin cell that have been methylated to be specialized to be a skin cell, for example. And if we remove this methylation, it will be open again and it will become a stem cell. So it could be specialized to anything afterwards. And then we can convert it back to a cardiac cell by methylating it at certain sites as well. So this opened the whole idea of converting cell types to other cell types. So basically we can convert your skin cells to stem cells, and stem cells, stands for a fetal cell, or your original very naive cell that could be redifferentiated to any other cell types. So this opened the field of induced pluripotent stem cells. I don't know how much you know about it, but this become, so this, we can take now a sample of your skin, convert it back to a stem cell, and then we can take these stem cells, convert it to cardiac cells, we can convert it to endothelial cells, we can convert it to kidney cell, to a liver cell, to any cell type that you want, according to a certain treatment of controlling how it's being methylated or demethylated. So this looks like a science fiction, but it's actually, we can do it now. So basically this is the process. So if we start all from a naive stem cell here, and this gets primed, so it can basically go either to ectoderm, mesoderm, or endoderm. And then we can go back this way. Now we can go back this way to any differentiated cell that we can go back and make it as a stem cell, and we can push it either ways. So we can push it to anything that we want. There was a major breakthrough happened in the past only 5 years or so that, I think my previous lab at the Gladstone Institute has led on this approach in the cardiac field, and this led to other discoveries in the other field afterwards. So it's called direct reprogramming. So all of the work before was based on basically pushing the differentiated cells back to stem cells and push it back towards other cell types. But can we push this one towards this one directly? I think this wasn't available at that time, so this happened in 2010, where my previous lab has discovered that we can push a fibroblast or a skin cell, or a fibroblast that exists in the heart, to a cardiomyocyte directly without going back to here, by using something called transcription factor. So the transcription factors are the proteins that are controlling the expression of the genes that you want on certain cell type. So basically, pushing a fibroblast with some cardiac specific transcription factor can push them towards cardiomyocyte fate. And basically this approach now has been developed in terms of IP and so on towards a company called Tamer Therapeutics. And this company, I was the first one in it 2 years ago, and now it's 35 people, and now they are taking it. So it's very successful in the translational models like pigs and so on. So now they are starting doing some clinical trials on it in treating heart failure. So this approach basically based on injecting the transcription factors that could convert fibroblasts into cardiomyocytes into the heart. So this will generate new cardiomyocytes in the heart. So this could support the failing heart and produce more power to eject blood. Because this is very translational, so I'm just going to talk about something that's very new, and this just been published this year. And this approach as well came out of the same principle. So the same principle was that the cardiomyocyte itself that's existing in the heart is not able to divide. So can we make this cardiomyocyte divide again? Can we make it like the young one that are able to divide? Can we reverse it back to in development? And how can we push it, right, to do this job? So we've done a lot of effort on this. And basically, this has been going on in the field for about 20 years. People have tried to use only single factor to push the cardiomyocyte to proliferate. But actually, none of these factors are being very successful, as you can see here summarized in this review article. And what you see here, just listed here, so these are the genes that have been used to induce cardiomyocyte proliferation or to make the cardiomyocyte again to proliferate. Like P38, neuroglin, urban NRG1, progestin, microRNAs have been used, YAB and cyclins. If you look to these markers, so these are markers for cardiomyocyte proliferation. They are called, so BRDU is a marker for DNA synthesis, for synthesizing DNA. PCNA is marker for DNA synthesis as well. Ki67 and phosphorestone H3 are markers for the cell being dividing. So these are expressed during the cell division. And if you see here to the numbers that people have been succeed with using single factors, it doesn't really exceed 1% of the cells that are able to divide again. So these approaches were not very successful, so none of these have become more translational or been taken forward. So what we did was pretty different. So we went back to the development, and we wanted to know we asked the question, so we know that the cardiomyocytes, when it's still in the fetal stage, they are able to divide. But when you get out of your mother's womb, you lose this ability. So what are the genes that are expressed in the fetal state that are not expressed in the adult state? And if we figure out a cocktail of these genes that could make your adult cardiomyocyte look like your fetal ones, so they will be able to divide again, and this will make your heart, able to heal after any injury, right? So we've done something called RNA-seq. So RNA-seq basically is a technique that quantify how many expression of each gene in the genome in terms of RNA. And we looked at the cell cycle genes that are highly expressed in the fetal heart and not being expressed in neonatal and completely absent in adulthood. As you can see here from this heat map, red means it's highly expressed and blue is very low expression. So we basically gathered about 35 genes, and we started to screen them to see what of these 35 genes we could combine, what was the minimal cocktail that we could combine and make the cardiomyocyte divide again. So we have used certain technologies for this. It's called high content imaging analysis to assess the proliferation of cardiomyocytes, and we use Adenovirus to infect these cardiomyocytes. And then what we found out is that there are 4 factors that are essential for this proliferation to happen. And these 4 factors are called CDK4, cyclin D, and CDK1 and cyclin B. So these are all 4 cell cycle factors. If you put them in cardiomyocytes, we get over 20% of the cardiomyocytes that are dividing. And as you can see here, these are different cell types. I'm not going to go through this in very details, but these are different cell types of cardiomyocytes. So these are mouse cardiomyocytes at day 7, isolated after 7 days of of birth. And these are adult rat cardiomyocytes. And these are, the green is the phosphostone H3, so this is a proliferation marker. And as you can see here, between the controls and the 4F or the 4 factors, if you infect with the 4 factor, you get really a robust increase in these proliferation markers, which is basically exceeding about 20%. We tested this in human iPS-derived cardiomyocytes as well. So these are the ones that I just talked about. We can push skin cells to become stem cells and then redifferentiate them to cardiomyocytes, and they are coming from humans. So these are called human-induced pluripotent stem cells, derived cardiomyocytes. And so these are human in nature. And as you can see, between the control and the ones treated with the 4F, they induce the proliferation markers very highly. So all of these proliferation markers are not indication to the actual splitting of the cells. So these are marking different stages of the cell cycle. But actually, we wanted to be more rigorous and basically investigate whether actual splitting of the cell is happening. So we couldn't do this with using the usual markers, so we have developed a lineage tracing mouse. I'm not gonna go through this because this is very complicated. But you know, this scheme of this mouse, of this genetic tracing mouse of proliferation, is the take-home message. If you find a green or a red cell, this means these are daughter cells. So the division happens, and you have daughter cells. So if you find a green or a red cell, this means you have a definitive splitting of the cell into 2 cells. Okay, so we've looked into this model, and we basically found, after injection of the 4 factors, we found many single-colored cells. These are the single-colored cardiomyocytes, as you can see here. And by quantifying them all total, so if you sum up the 8 and the 10%, so this is about 18 to 20% in total of the cells that of the cardiomyocytes are actually divided. We've done some assessment to see whether this gene therapy, if we inject these 4 factors or 4 genes into the heart, whether this could repair the heart failure or not. And what we've done, we induced myocardial infarction in mouse heart. So basically it's like you get a heart attack, so we induced this in the mouse heart. And then after 7 days, we injected viruses encoding these factors. And then after 12 weeks, we have done MRI and histology. And as you can see here, these are the control hearts. So you see that the amount of damage is really high, which is basically this blue thing here. This means these are fibrous tissue. So this is what you get after a heart attack. So basically your muscle converts to a fibrous tissue like this. But if you inject these 4 factors, you get more muscle around supporting this. So you see, if you look here, this still a blue thing, but it's been supported by more muscle because we are producing more heart cells in here. So this supports basically the heart, and this was very promising in basically improving the ejection fraction and put it back almost to normal level mouse. So this was very promising, but you know, using 4 genes is not really translational. It's too much to inject 4 genes into human. So we went and tried to find out whether we can reduce the number of genes somehow. And we did a small molecule screening. My initial degree is in pharmacy, so I usually do a lot of small molecule screening, so because I still like it. So we found that when we add these 2 small molecules, it's called MK1775 and SP431542, with only 2 of the genes, we get similar proliferation amount that we get for the other 4 genes. So this was very interesting. We've tested this to see whether we get single, green or red cells as well in this lineage tracing mouse, and we found that we get similar amount of these single cells. And we've done as well assessment to see whether we could improve the heart function using these 2 factors and 2 inhibitors or 2 small molecules. And basically, we got this very similar result to what we got with the 4 factors. So this was very encouraging. And basically, this cocktail basically could be a bit translational. We're still working on this a bit in terms of translation. But I think what's really exciting about this project is that this cocktail of 4 factors is not only able to make the heart get better, it could act on other tissues. You know, so for example, for the spinal cord injury, you lose a lot of neurons and you want to regenerate these neurons and nobody's able to regenerate these neurons. In diabetes, you lose a lot of your beta cells in the pancreas, and basically we are not able to regenerate these beta cells so far. So in collaboration with some other groups, we've tested the ability of these 4 factors to make the neurons divide in culture. So this is a neuron, a single neuron that they label with a reporter called MAP2 GFP. So the MAP2 is a specific protein that's expressed only in the neurons. So being green means it's a neuronal cell, and you can see the dendrites and so on. And basically they did time-lapse imaging over time. As you can see, this single neuron starts to divide. It makes 2 cells, and these 2 cells makes 4 cells, and these 4 cells start to divide again. So basically these 4 factors basically are able to increase the division of neurons. So this will have an impact as well on something like spinal cord injury. And this is the quantification basically, so they've done 12 replicates on this. The controls, they haven't seen more than 1 division in any well. But treated with the 4 cells, they have seen up to 12 cells are performing at least 4 or 5 times divisions. So, and basically what they label totally in the whole cell, in the whole well, is not more than 40 cells. So this is about 25% of the actual cell number in the well. And we've tested this in collaboration with people who are working on pancreatic islets as well. So these are pancreatic islets from mice, and they are treated with control virus or with the 4F. And the green basically is a marker for DNA synthesis or for cell proliferation, and it's called EDU. So basically, as you can see here, the 4F really induced a lot of cell division in beta pancreatic cells. So this could have another potential in treating, diabetes as well. So just to summarize this part, we have found that a cocktail of 4 factors could induce a full cell cycle or induction of cardiomyocyte proliferation, and this could help the heart to heal after injury. And we've done some mechanistic studies on this, like some inhibition of some other components of the cell cycle and so on, which I think was very promising as well. So currently my lab is focusing on making this more translational. So we are, because injecting cell cycle factors into human is very dangerous, because this could spread all over the body. It could induce some cancer or something like this. So we are trying now to control the expression only in the heart under certain promoters. So we can do that, and we are testing this idea at the moment, and we are making as well the expression very controlled in time. So there are some tools, there are some viruses that doesn't last for a long time, so it lasts only for 3 or 4 days. So it will give it a shot, increase the cardiomyocyte proliferation for 3 or 4 days, and that's it, and it will disappear from the body. And that's what we are working on right now. So I'm gonna switch gears from this hardcore biology thing to something probably more relevant to you guys, which is probably biomimetic culture and tissue engineering, more or less. So, but this basically came from my interest in testing these therapies in humans. So to test a gene therapy in human, I think it's impossible really, unless you conduct a full clinical trial. And you don't really want to go to a full clinical trial on it unless you are really sure that people will not get harm, or you're not really, and you get really reasonable benefit which will justify you are going to a clinical trial. So we wanted to get human hearts, a tissue that's similar to the human heart, but culture it in situ and try to test, therapies, therapeutic effects on in situ in a dish. So we found that, there's a technology that's been developing over the past 10 years or so that's called heart slice culture. So you can basically culture heart slices of 300 micron, and people are trying, this is how people does it usually. So they get a pig or human heart, and basically they cut it using a vibratome while it's viable, and then they lay it over a transwell, and this transwell is basically a transmembrane, and it's exposed to the media from the bottom, and it's exposed to the air from the top. So the idea is that this heart slice will absorb the media component from the bottom, and the top is oxygenated, so it's not hypooxygenated. However, this method, nobody has been able to keep the heart slices for more than 24 hours. And 24 hours is just not enough to test any therapy or any effect of anything really. So we thought about how could we make this better? So we thought about how the heart is and how these heart slices are doing inside the heart. And we should really mimic all the conditions in the heart in this dish, right? We can't just leave them like this and tell them, do well. It's just nonsense. So we've tried to mimic all the conditions in the heart. So if you think, what are the conditions in the heart? So the heart is under continuous stimulation, electrical stimulation. So basically there is induction of electrical signal, which happens at 1.2 hertz, so it's 72 times a minute. And there are coronary arteries feeding this heart and providing oxygen to it. The heart tissue is freely moving, so it has to have the ability to really move and contract. Otherwise, it's just going to be deformation happening. So what we've done, we've developed a system to oxygenate the heart and to stimulate the heart as well with this. So this is a 6-well cover, a 6-well plate cover that has got 2 graphite electrodes at each end of the well. And basically this could be connected to a stimulator, and this stimulator could induce electrical stimulation at the beat rate that you want and the voltage that you want, and put the current that you want as well. So that's how we do this. So basically we have the plate cover, the heart slice, we have it, so we basically have to put it on a support like this, so it's becoming, freely hanging in the media. And basically we support it here, and then we put it in the 6-well like this, and then we put the cover on the top, and then we start the electrical stimulation. And to assess the viability of these tissues, we use something called MTT assay. So this is a viability assay that's depending on how good the mitochondria function is, or the electron transport chain is in the cardiomyocytes. So what we found is that using the currently published protocol, either with the stimulation or without stimulation, these doesn't keep these heart slices over 24 hours. So we started to do screening for the media components and so on, and we ended up by finding an optimized growth media for these heart slices, and we optimized all the culture conditions under oxygenation, the stimulation rate, the stimulation voltage, and so on. And under these conditions, we could keep these heart slices fully viable and exactly similar to the uncultured heart slices for full 6 days. So 1 week of full viability, and this should be enough for you to test any therapeutics, basically. After 6 days, they start to decline very rapidly, and basically they reach about 50% of viability at day 12 after culture. But I think we are very happy with these 6-day window that we could test, immunotherapeutics. Usually we need 2 to 3 days, so 6 days is really more than enough. So this, I'm just showing you how the heart slice look between day 1 and 6. So usually the heart shouldn't beat until you electrically stimulate it. So here, yeah, so all of this is still unstimulated, so we didn't stimulate it, so it doesn't really beat anyway. But once we stimulate, you start to see the calcium. So green means the calcium is basically flashing in these heart slices. And they are very synchronized, so they synchronize very well with the stimulation. So once you stimulate it, you start to fire calcium. But at day 7, we start to see these cells, which are basically spontaneously releasing calcium, and that's basically a sign of starting deterioration of the heart slice. But still, the heart slice itself still responds to the calcium, still responds to the stimulation. But by day 10 and 14, you can see here, it's so you see it's completely out of control. The calcium is completely out of control, and they just go really bad after this at day 14. So at day 14, even they are not carrying any more calcium in there. So we've been able to assess, more in depth the functionality of these heart slices over time, and we've done some contractile measurement in collaboration with Dr. Ashok Kumar in the anatomy. And basically, what we do here is we hang the slice here between forced transducer and a stable puller here. And basically, there are 2 electrodes here, and when we stimulate electrically the slice, it starts to contract, and you can see here this is the contraction. And basically, we could assess, the software can assess how much force it's been generated during this contraction, and what's the relaxation rate by measuring the time to decay. As you can see here, this is data from day 0 compared to day 6 heart slices. So the day 6 heart slices are fully viable still. They are very compared to the day 0. If you stimulate with isoquinoline, you get a good inotropic effect. So it's basically inducing the contractility. And basically you get as well increased speed of the relaxation, as assessed by the time to 50% and 100% relaxation parameters as well. We've done something that's to get sure that these heart slices are fully viable for 6 days. So we looked at the whole gene transcription in these heart slices using RNA sequencing. So this RNA sequencing is measuring how much transcript of each gene, of about 16,000 genes, are expressed in these heart slices. So being not changed means there's no major changes in these heart slices. So as you can see here, if we compare, so this is the full genome, so all of these dots are, each dot basically is a single gene. And this is the p-value of it changing, so if it is this minus log 10 to the p-value, so this p-value of 0.05. So if you compare day 2 of culture to day 0 means it's uncultured slices, and there's only 2 genes that been out of the 16,000, so it's not really significant. If you look at day 6, we have about 5 genes, But if you look at day 10, when they really deteriorate, there's over 500 genes that have been changed. So I think this is basically very conclusive to that our heart slice culture is very good for full 6 days with no major changes in the nature of the heart tissue. This is just to look at the expression of certain cardiac and metabolic genes and fibroblast genes, and as you can see, in day 10 we get a high down regulation of the cardiac genes and high up regulation in the fibroblast genes, which means that the cardiomyocytes are dying in culture and then the fibroblasts are taking over the culture, basically. But this was very interesting. We looked as well to certain, the connexin 43 is very important protein in terms of cardiac conductance, and basically it sits between each cardiomyocyte and the other one. So this protein is very interesting for the conduction of any electrical stimulation. So we looked more specifically for this gene, and because we are not looking only for the expression, we are looking for the position of this protein. So this protein has to basically sit in the junctions, like what you see here in the control uncultured slices, in the junctions between the cardiomyocytes. And this gene if you do the stimulated culture at day 6, it sits in the right position actually, and still expressed in all of these cardiomyocytes. However, so this comparison just as a negative control to show you how the deteriorated culture looks like. So this is day 6 of unstimulated culture, which basically deteriorate after 24 hours. And you can see here, so it disappears completely from here, from this region where there's actually cardiomyocytes. It becomes very dotty and so on, so it's not anymore like lines, so the conduction basically is deteriorating. So I think this was very interesting to test whether we can actually test our therapies in this. So we can test the effect of these 4 factors that we think that they can induce cardiomyocyte proliferation in human heart slice in situ or not. And basically, as you can see here, so this is an unstimulated culture and this is the stimulated culture that we optimized. And if we infect with a GFP, so GFP is a green fluorescent protein, so if we infect with a GFP virus, you get really very good expression on most of the cardiomyocytes in the stimulated culture only, which we have optimized. So this was really a major reason that we wanted to optimize this culture. And we were very happy that it served our cause, basically. And here, this is showing that the effect of the 4F, so we can basically induce some proliferation markers in these heart slices. As you can see here with the 4F, so this is called phosphorestonage 3, so this is one of the proliferation markers. And as you can see here, this nuclei is co-stained with phosphorestonage 3, and it's basically a cardiomyocyte probably in a state of division still. That's been stained with this marker for the cardiomyocytes. So I think we want to make this even better and live longer in culture. So we are collaborating with Dr. Guru here, and basically we are trying to put our slices under mechanical stimulation as well as electrical stimulation. So what we've done in our culture system is that we developed only electrical stimulation. But
0 to about 120. And diastolic pressure is approximately between 5 and 10 millimeters of mercury. Pressure is approximately 120 millimeters of mercury. And here's the whole setup. Along with a systolic fraction of 40% and 70 beats per minute.
Okay, so I think, so this is really exciting. We are very excited about this collaboration, and we are hoping that, this could keep them for a month or something in culture viable, because I think we are lacking in our system. I believe we are producing the electrical stimulation, but still the mechanical stimulation and the stretch. So this is very important, I believe, element that we couldn't provide before, but I think now with Dr. Guru, we are starting to test this hypothesis, and it's very promising. So, yeah. Okay, so just to conclude, so now we are able to develop a human or big heart slice culture system that's fully viable for 7 days. And the culture system is a valuable tool for efficacy, to test the efficacy of any heart failure therapy in the future. At the end, I would like to thank the team in Tamer Therapeutics. Most of this work being originated there, and my lab, my current lab, and AHE and NIH for the support for this work. And thank you so much.